Chapter 16 deals with the molecular basis of inheritance. In this chapter, we're going to look at some experiments that showed that DNA is the genetic material. We're going to look at how many different proteins work together in order for DNA to replicate itself. And we're going to look at how a chromosome consists of a DNA molecule packed together with proteins. We're going to go over the background of one experiment here, and in the next slide, I'll detail for you the different steps. So the discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. At this time, it was not known if DNA or proteins were the genetic material. There was a little bit more support for proteins at this time because proteins have so many different functions. Griffith worked with two strains of bacterium. One strain was pathogenic. This was represented by the letter S. This was because it produced a smooth capsule around itself, S for smooth. The capsule was able to protect the bacterium from the organism that it was invading. The other strain was harmless. This was represented by a letter R. R stood for rough because it didn't make a capsule. Therefore, an organism that was infected by the bacterium could kill the bacterium. So there's a pathogenic strain, S, and a harmless strain, R. And he found that when he mixed heat-killed remains of the pathogenic strain with living cells of the harmless strain, some living cells became pathogenic. He called this process transformation. This is when there's a change in genotype and phenotype due to the assimilation of foreign DNA. Now we're going to look at the different steps of the experiment. In the first group of mice, he injected them with living S cells. These were the pathogenic bacteria. As expected, the mice died. He then injected a group of mice with living R cells, the non-pathogenic bacteria. These mice lived. He then injected a group of mice with heat-killed S cells. Now these were pathogenic bacteria, but because they were heat-killed, they were no longer pathogenic. They were non-pathogenic. These mice lived. All of these results were as expected. The interesting result was in the last group. He mixed some of the heat-killed S cells, which we just saw let the mouse live, and living R cells, which we saw before also let the mouse live. When he put these both together and injected them in the mouse died. So he found out that something was making the living R cells living S cells, and he called this transformation. And what was happening is that DNA from the S cells was being taken up by the R cells. Now, one thing I want to make clear from that previous experiment is that his research didn't actually show that it was the DNA. We now know that it was DNA, but his research led to further research that helped show it was DNA that was being taken up. Some of that further research was done by researchers, one group of them called Avery, McCarty, and McLeod, and they helped identify that this transforming substance was DNA. Now, many biologists still remain skeptical, mainly because we didn't know much about DNA back then. But there is more evidence for DNA as a genetic material from studies by Hershey and Chase. And they did their studies with viruses that infect bacteria. Viruses that infect bacteria are called bacteriophages. Now, a virus is an infectious particle of either DNA or RNA enclosed by a protective protein coat. It's not capable of replicating outside of a cell. So a virus has to enter its cell so that it could replicate. Figure 16.3 shows what a typical virus looks like, and it's showing it infecting a bacterial cell. So there's a head structure. They call that the phage head for bacteriophage head. There's DNA. In this example, it's DNA that's in it. There's a tail. So it's a very simple structure, a virus. Now, there are two findings by a researcher named Erwin Shergath that became known as Shergath's rules. And these had to do with the structure of DNA. So one thing that he found was that the base composition of DNA varies between species. So for example, let's say in humans, there's 20% of the bases are adenines. Maybe in mice, it's 10%. So the base composition varies between species. The same bases are involved, but there'll be different amounts of them. Another finding that he had was that in any species, the amount of adenines was approximately the same as the amount of thymines, and the amount of guanines was the same as the amount of cytosines. Now, the basis for these rules was not understood yet until the discovery of the double helix. But because we know this, we could predict the amount of bases if we just know the percentage of one base. So for example, if I were to tell you 10% of the bases were adenine, you should be able to tell me that that means that 10% of the bases in the organism are thymine, because there's equal amounts. You should also be able to tell me that there's 40% guanine and 40% cytosine. Now the way we get that is we know it has to equal 100%. If 10% are adenine and 10% are thymine, that means there's 80% left. C and G have to have equal amounts, so we take that 80 and divide it by 2 to get 40% of each of those. Now also contributing to identifying the structure of DNA were Maurice Wilkins and Rosalind Franklin. They used a technique called X-ray crystallography. And what Franklin did, she produced an image of the diffraction pattern of the DNA molecule using this technique. And this let Watson deduce from Watson and Crick that DNA was a double helix. He also was able to figure out the width of the helix and the spacing of the nitrogenous bases, so how far the bases were from each other. The picture in the center is Rosalind Franklin, and the picture to the right of her is that X-ray diffraction pattern. This is called photo 51. Now at first, Watson and Crick thought the bases paired like with like. So for example, they thought A paired with A, T with T, etc. But that didn't produce a uniform width. They found instead that pairing a purine, which are adenines or guanines, with a pyrimidine, which were cytosines or thymines, resulted in a uniform width consistent with the X-ray data that was obtained by Franklin. This model that they developed, Watson and Crick, also explains Sargaff's rules. It explains why the amount of A was equal to the amount of T, and the amount of G was equal to the amount of C. This slide is showing how they knew it had to be a purine binding to a pyrimidine. So when they did the X-ray crystallography, they got a certain width. If they had two purines bound together, the helix would be too wide. Two pyrimidines would be too narrow. So it had to be a purine and a pyrimidine to be the correct width for the DNA double helix. We're going to move on to replication. Since the two strands of DNA are complementary, each strand acts as a template for building a new strand. In DNA replication, the parent molecule unwinds and two new daughter strands are built based on base pairing. So if you look at panel A, we have the parental molecule, we have the two strands of DNA. In B, those two strands separate from each other. And you'll notice that these original parental strands, the bases are in dark blue. Then for each strand, complementary bases are going to come in and base pair with the bases that are exposed. And they're going to be represented by light blue colors. So for C, you see there's a dark A, a light T comes in. There's a dark C, a light G comes in. And that is also what's happening on the right-hand side. So we get two strands of double-stranded DNA. Now the model that explains DNA replication is called the semi-conservative model. And I just want you to focus on the first part of the picture. So we have two strands of DNA. And what the semi-conservative model states is that when a double helix replicates, each daughter molecule will have one old strand and one newly made strand. The end part of that picture shows if those two then replicate. But again, the semi-conservative model states that after DNA replication, one old strand is paired with one new strand. There were some other models that they tried to explain it with, but those were not followed. So we're just going to focus on the correct one, the semi-conservative model. Now we're going to look at DNA replication in a little more detail, and we're going to focus on E. coli. More than a dozen enzymes and other proteins participate in DNA replication. Replication begins at particular sites. These are called origins of replication, which are short sequences of DNA. This is where replication begins, and it's where the two DNA strands initially separate from each other. They form what's called a replication bubble. Prokaryotes, such as E. coli, have one origin of replication. A eukaryotic chromosome may have hundreds or even thousands of origins of replication. Replication proceeds what we call bidirectionally. It goes in both directions from the origin of replication until the entire molecule is copied. Now, figure 16.12a is showing the origin of replication in E. coli. So if you start with the top left picture, we have double-stranded DNA. Remember, in prokaryotes, the chromosomes are circular, and it's pointing at an origin of replication. As replication proceeds, at the origin of replication, the two strands start to separate. As they separate, what's called a replication bubble forms. The actual place where they're separating is called a replication fork. New bases come in complementary base pair to where it's being separated. And then we end up with two new daughter DNA molecules. One thing to note here is that the template strand is called the parental strand, and the newly made strand is called the daughter strand. That has nothing to do with sex. It's just called daughter because that's what it's called. It doesn't mean that it's female. Now in figure 16.12b, we see origins of replication in eukaryotes. So like we said, eukaryotes, they have multiple origins of replication. That's because they have a lot more DNA. So they need more places for it to start in order for it to finish accurately in a timely manner. So if you look at B, we have several origins of replication. The strands separate and replication proceeds in both directions, basically until those bubbles meet each other. And eventually there'll be two daughter DNA molecules. Each one will have an old strand or parent strand, and each one will have a newly made daughter strand. So what happens during this process of DNA replication? At the end of each replication bubble is a replication fork. This is a Y-shaped region where new DNA strands are elongating. A group of enzymes called helicases are important. These are enzymes that untwist the double helix at the replication forks, separating the two parent strands. Another group of proteins that are important are single-strand binding proteins. These bind to and stabilize the single-stranded DNA. They make sure that when the helicase separates the two strands, they don't come back together. We don't want that to happen. We want new bases to come in. Topoisomerase is an enzyme that relieves the strain of twisting of the double helix by breaking, swiveling, and rejoining DNA strands. DNA polymerases catalyze the synthesis of new DNA at the replication fork. So basically, as the new bases are coming in, it's the DNA polymerase that actually forms the bond between the existing base and the base that's being added. Now, DNA polymerases, interestingly, aren't able to start the process themselves. They require a primer to which they can add nucleotides. The enzyme that makes this primer is called primase. And in figure 16.13, we see these proteins that are involved in the initiation of DNA replication. We have that topoisomerase enzyme that relieves any strain from the twisting. We have the primase that makes that initial little primer so that polymerase can do its job. We have the helicase, which is shown in green, which is separating the two strands. And we have those single-strand binding proteins, which bind to either of the single strands so they don't come back together. So in figure 16.14, we see the addition of a nucleotide to a growing DNA strand. If you look at the left-hand side of the slide, we have a new strand and we have the template strand. On the template strand, an adenine is open to be bound to. So what's going to bind to that? A thymine. As each monomer joins a DNA strand, this occurs through a dehydration reaction. It's going to lose two phosphate groups. Now it's important to note that DNA polymerase can only add nucleotides to the 3' end of a growing strand. It cannot add to the 5' end. It has to add to the 3' end. Now this next slide is going to be a little bit confusing. So just look it over a few times. You may want to try Googling a video on it. So when DNA replication occurs, because the strands are anti-parallel, there's actually what's called a lagging strand and a leading strand being made. The leading strand, which it's pointing at the arrow in that diagram, is made continuously, and it's made toward the replication fork. So as the two strands separate from each other, nucleotides come in and are added. The lagging strand, however, is made in fragments away from the replication fork. And again, the reason that is because nucleotides can only be added onto the 3' end of the growing strand. So it opens up a little, some DNA is added. It opens up a little more DNA is added. These little fragments that it's made in are called Okazaki fragments. And these are going to end up being joined together by DNA ligase. So there's a leading strand and there's a lagging strand on both parent strands that are being made. Figure 1615b is showing the synthesis of the leading strand and some of those different proteins that we talked about. So we have our parental DNA being separated. You can see we're on the right-hand side where the origin of replication is. The helicase is in green. That's separating the two strands. The specific DNA polymerase is called DNA polymerase 3, and that's moving down the DNA strand, and it's adding nucleotides to the 3' end of the growing strand. We also see there the single-strand binding proteins that keep the strands separated so those no nucleotides can be added in there. We also see at the very beginning of the newly made strand that RNA primer. So if you remember, DNA polymerase cannot begin synthesis of a strand. It has to have something that it could start building onto. And in the bottom part of the slide, it just shows that continuous elongation in the 5-3 direction. In figure 16.16b, we see synthesis of the lagging strand. So primase makes the RNA primer. DNA polymerase 3 comes in and makes the first Okazaki fragment. As it opens up more, another primer is made and another Okazaki fragment is made. And this process is going to repeat itself over and over again. This is just showing you continuing. Again, for each little Okazaki fragment that's made, another little RNA primer has to be added first. DNA polymerase 1 is going to replace that RNA primer with DNA. And like I mentioned before, DNA ligase forms the bonds between those DNA fragments. And again, the overall direction of the strand being made is in the 5' to 3' direction. Now this slide, this is a complex slide. This is just showing you everything happening at once. synthesis of the leading strand and synthesis of the lagging strand. And they sometimes call this the trombone model due to its shape. DNA polymerase has the ability to proofread, which means it could go back and see if there were any wrong bases, for example, that were added and fix it. It's very good at this, but it's not perfect. So sometimes there can be a change or a mutation, and this can become permanent and be passed on to the next generation. These mutations are the source of genetic variation that natural selection operates on. These are what are ultimately responsible for the appearance of new species. Now, DNA polymerase does have some limitations that can cause problems when replicating the linear DNA of eukaryotic chromosomes. So these limitations we mentioned before, but we didn't call them limitations. One is that a primer is needed to start the process, and a second is that DNA polymerase can only add nucleotides onto the 3' end of the growing strand. So this means there's no way to complete the 5' ends. So if there were repeated rounds of replication, it would produce shorter DNA molecules with uneven ends. Now this isn't a problem for prokaryotes because prokaryotes have circular chromosomes. So in order to fix this, eukaryotic chromosomal DNA molecules have nucleotide sequences added to their ends called telomeres. Now these don't stop shortening, but they do postpone it. The shortening of telomeres is connected to aging. So the older a person is, the shorter their telomeres are. If chromosomes of germ cells became shorter in every cell cycle, essential genes would eventually be missing from gametes they produce. The enzyme telomerase catalyzes the lengthening of telomeres in germ cells. Figure 16.20 shows how over time the ends of the linear DNA molecules shorten. Now, the last concept that the book talks about is how a chromosome consists of a DNA molecule packed together with proteins. So in order for DNA to fit inside a cell, it has to be compacted. In a bacterium, the DNA is what we call supercoiled. It's found in a region of the cell called the nucleoid. Remember, prokaryotes don't have a nucleus. In the eukaryotic cell, DNA is precisely combined with proteins in a complex that is called chromatin. In eukaryotes, there's a more complex system of packing involved in order to get this chromatin to the right size. The first level involves proteins that are called histones, and they're responsible for the formation of what are called nucleosomes. This is a section of DNA that's wrapped around 8 histones. It kind of looks like the beads on a string. Nucleosomes are important in regulating gene expression. Now in figure 16.22a, we see a double helix, and then further down on the right, we see a formation of a nucleosome. It's the DNA in blue that's wrapped around these special proteins called histones. The histones are the little purple circles with their histone tails. And again, it wraps around 8 of those histones. And as you can see where it says nucleosome, if you look at it under a microscope, it kind of looks like beads on a string. The next level of compaction is the formation of the 30 nanometer fiber. This is formed from interactions between a histone called a linker histone and the next nucleosome, so the DNA in the next nucleosome. So basically, it's a histone binding to DNA that's just going to pull those nucleosomes closer together. The next level of compaction after that is the formation of what we call looped domains. This is when the 30 nanometer fiber folds and attaches to the nuclear matrix. Again, we're pulling things closer together, making them more compact. Most chromatin is loosely packed in the nucleus during interphase and condenses prior to mitosis. Chromatin can vary in how tightly it's packed together. Euchromatin is what we call chromatin when it's loosely packed together. Heterochromatin is what we call chromatin when it's tightly packed together. The dense packing of the heterochromatin makes it difficult for the cell to express genetic information that are coded in these regions. Figure 16.22b illustrates this next two levels of compaction. So first on the left-hand side, we see the formation of the 30 nanometer fiber. And again, this is when the nucleosomes are pulled closer together. After that, we see the next level was the loop domain. And this again is when the 30 nanometer fiber attaches to the scaffold, or what we call the nuclear matrix.